Alright, so today we need to go ahead and start talking about the scientific method. Next class we are going to start our enzyme lab, but you need to know why we do this stuff in the first place. So let's go ahead and talk about the scientific method. How is science done? Well, science always begins with some observation that we make. Our observations are going to be through the five senses. It is sight, touch, taste, feel, and hear. So what is it that we're seeing? What is it that we're smelling, that we're feeling? So this is the process that we gather the information about events and processes in a careful, orderly way. We have to be able to gather our data and all the things that we observe. And so that brings us back to the differences between our quantitative and qualitative data. Quantitative, the N is in there, the Q-U-A-N, quantitative, that means we are dealing with numbers. Those numbers are obtained by counting or measuring. But then you have qualitative data, and that's more of the description. It's telling us about the quality of something, how it looks, how it smells, the things that can't be put down in numbers. So that's quantitative data versus qualitative data. So we're going to start off with a hypothesis after our observation. Remember, we've seen things, we've done a little research, and now we're going to hypothesize by it, which is really simply saying is a scientific explanation for a set of observations. It is our educated guess. It's why do we think this happened and what's going to happen because of it. A hypothesis must be stated in a way that makes it testable. The hypothesis is just a possible answer to a question and it must be thoroughly tested. A lot of the times you'll see your hypothesis being if so and so happens, then this will happen. And so it's giving us something that we can test with if this, then it's giving us our data to collect, then this will happen. So the scientific method is a series of steps used by scientists to solve a problem, answer a question. We observe, we ask a question, why does this happen? What's going to happen when we put enzymes in with a substrate? Then we're going to form a hypothesis to kind of say, we know this is how it's supposed to work, and then this is what we're going to see. Then we're going to design a controlled experiment, and this is important to be a controlled experiment because too much happening totally skews everything and we're getting lost in it. Then we're going to record and analyze our results and finally draw conclusions to see what all this lab activity and everything has told us about this subject for us is enzymes. So what is it going to tell us that we can draw from this experiment? So step one, observe, ask a question. So a problem or question must be first identified. How much water can a root hair absorb? Well, okay, so we're actually looking at things in ways that we can measure, remember? So we would be able to kind of gather how much or record how much water would be absorbed. Why does a plant stem bend toward the light? Or what effect does temperature have on heart rate? So all these things are something that we see happening, and now we want to know why it happens. Then the hypothesis follows that it's a possible explanation to the question. It's simply a prediction. It's not yet been proven. So here it is. It must be stated in a way that is testable. Remember, it has to be testable. That's what the lab is all about. A statement considered testable if evidence can be collected that either does or does not support it. Okay, that means we have to test it to see if our guess is right or if it's not. And then we're going into step 3. And this is where it gets a little more difficult. I would say that variables come into play, and we'll go over this a little bit more in another video. But let's talk about a controlled experiment. The factors in an experiment that can be changed are called variables. And so let's say going back to the temperature and the heart rate on that last slide, well, those are things that can be changed and can be recorded. Some examples of variables would be changing the temperature, the amount of light present, time, concentration of solutions used. Remember, it is all testable, so we're able to measure it, we can record it, observe it. And so a controlled experiment works with one variable at a time. That is key. One variable to change at all times. That means I am in charge of something and I am going to change that one variable. If several variables were changed at the same time, then you don't really know what is really happening or why is it happening, because you got different things happening. I mean, if you have a temperature change and you have sunlight in there and you're judging both of those, and then you want to pull in the growth, you don't know if the growth is really because of the temperature or the growth is because of sunlight. It's why we have to keep it one variable at a time. So in a controlled experiment, only one variable is changed at a time. Like I said, all other variables should be unchanged or controlled. That's key. A controlled experiment has a controlled variable. An experiment is based on the comparison between a control group with an experimental group. The way I kind of like to say it, a control group is the thing that's going to stay normal. It's the thing that doesn't get messed with at all. So if we have, again, playing with the different growth in plants, well, we're going to stick a plant in a normal atmosphere, a normal environment, don't mess with it, and then we're going to take plants and we're going to play with things, to play with the temperature or play with how much light, and those are the experimental groups, the ones we're changing things with, and the other got nothing done to it is our normal, is our control group. So like I said, reading on, it says two groups are identical except for one factor, that controlled variable. The control group serves as a comparison. I repeat, we have to have a control group so that we can compare and see if the lab is actually working, see if our experiment is actually measuring something or seeing something happen. It is the same as the experimental group, except that one variable is being tested, is being removed. The experimental group shows the effect of the variable that is being tested. So, control group gets nothing done to it. Experimental group is played with, done something with, we're experimenting on it. So, that is our experimental group. But we're going to take it and compare the results from the experimental group to the control group to see if we're really making a difference. But it is important that you guys know these two words, placebo and bias. A placebo is a substance that is made up to resemble drugs but not contain an active drug, but is made of an inactive substance such as starch, sugar, blah, blah, blah. Okay, what you need to kind of realize, a placebo is something that's really going to trick our brain. If we have, we want to test a drug, like this is saying, and then you have one group that is getting a white pill, and then you have one group that is getting no pill. Well, they automatically, the group getting no pill knows that they're not getting that medicine, and so they're automatically going to think, okay, we're not getting it, go on. We can't experiment. We can't see what's going on because they know they're not getting the medicine. But if we give group 1 the drug and the white pill, and then we're going to give group 2 a drug or not a drug, but a white pill that is really just sugar, then they do think that they're having something. They think that there's a possibility that they are getting the medicine. And so the brain is going to trick their bodies just enough to kind of keep the experiment rolling. But by the end of the experiment, you can tell the big difference between who has the drug and who is not. One thing I almost like to bring up here is that there's such a thing as a fake pregnancy. And this is kind of the brain having one of those placebo effects. It's a circumstance where, and I actually saw it on a show not too long ago, where a lady had a tumor, but her body had tricked her into thinking she was pregnant. And so she had all the symptoms, the nausea and the cravings and weight gain and everything. But it wasn't a true pregnancy. Instead, the tumor had tricked her body into thinking that she was pregnant. So it was a fake pregnancy. And that's kind of what a placebo does. It is just enough to give your brain that, hey, this might be happening, but then it's going to take it a step further and allow you to see how much the drug is really affecting and how much it is just your mind playing tricks. That's the placebo, the sugar pill in it. And then bias is a prejudice in favor of or against one thing, a person, group compared with another. Kind of think of it this way. I think that my daughter is absolutely one of the best ball players around. She's a catcher. She's a beast. She can throw down like nobody playing. I mean, she's awesome. But that's my bias. Not everybody else is going to think that my daughter's as great as she is. That's the bias, the rose-colored glasses. I think I like chocolate ice cream, so chocolate ice cream is the best. Anytime I get around other ice creams, I'm going to go for chocolate. That's the bias, is my preference over something else plays into effect. The example says using friends as test subjects, offering compensation for agreeing with you. I mean, you're either paying them off or they're not going to say no to you because they like you. And that's the bias. It's kind of like you football players and people into sports. Well, you get some of these refs who aren't calling the game right and they're kind of going towards one team over the other. Well, it's probably because the ref is more biased to that team. And so that's the difference. Placebo is your brain tricking you into something, but you give them a sugar pill instead. Bias is already leaning towards something, and so you're going to go ahead and automatically go with that side. So we already talked about the control group. We talked about an experimental group. But we need to talk about going into the other, what variables are kept constant, what variables can be changed. Here's our experiment. In order to test effectiveness of a new vaccine, 50 volunteers are selected and divided into two groups. One group will be the experimental group, and the other will be the control group. Both groups are given a pill, that's the placebo, to take that is identical in size, shape, color, and texture. But remember, only one group is getting the actual drug, the other group is getting the sugar pill or the placebo. Even though the volunteers are given identical looking pills, the control group will not actually receive the vaccine. So the control group doesn't get the medicine. The experimental group is the one getting the medicine. The variables that are kept constant, so they're either called constant or control variables. That's the size, shape, color, and texture of the pill. What variable is changed? What am I changing as the experimenter? The experiment. Yes, I don't know what word I'm going with. The scientist. Whatever. Whether or not the pill contains the vaccine. All right, so we have to break down the experiments like this, and you will have questions coming at you on test that's asking what's the control, what's the experiment, independent variable, dependent variable, all that. So you've got to be able to break down an experiment when given this. So let's get into independent variable. Is a variable deliberately changed by the scientist? That's why I keep saying I change it. I am changing it. I am an independent woman, so I change it. I'm in control of changing what I want to do in this lab, and so that is going to be the independent variable. Now, the dependent variable is the one that is observed, or you see what happens to it because of the independent. This is where you collect your data from, so that really you can think of it like this. The dependent variable is going to be the thing that is recorded, observed, measured, or counted. Those are the main things that come up over and over again. We observed this happening, they recorded this, they counted the number, and they observed, recorded, measured, and counted. You get the point. So independent, I change it. Starts with an I. It is independent. I change it. Dependent depends on the other. So we get to record, measure, count, observe what is happening, and then get the data from it. The data has to be collected and must be organized and analyzed to determine whether the data are reliable. Does it support it or does it not? It's important to get accurate accounts of your data because you want it to be exact measurements. You want it to be as precise as you can so that if some other scientist comes in, they can redo the exact lab and you might come up with some similar data if you've done it correctly. And then finally, you want to draw conclusions. The evidence from experiment is used to determine if the hypothesis is proven or disproven. So experiments must be repeated over and over. You can't just do it once. Okay, granted, in class we will probably only do it once because that's all I got money for or time. But most of the time, experiments will be done over and over again, repeated again and again, at least 3 times, just so that we can make sure we have a valid amount of data and they're all pretty much the same. If you have something like the first 2 trials, it goes, you get 10 of this and 10 of that, and then all of a sudden on the third one, you get 100, you probably know that was a screw up. And so you're able to kind of see if things went wrong or did not, and we can validate the lab itself. And so I'm going to go over the worksheet problems in the next video. You can go over these several experiments on your own with the PowerPoint online. It is in the scientific method module. All right, well, we'll see you next time.